What I'm going to be talking about is the what made us as Christian Hansen go on this journey to launch the first wine bacteria exclusively for bioprotection. Cool. So I've actually stolen this picture from Professor Coquelin's group. It's from a recent literature review. The reference is on the bottom. I actually urge anybody interested in the topic of bioprotection to read this. It's an excellent, excellent review. And up till now, we've very much, as an industry, focused on biological protection in wine exclusively around yeasts. And Professor Coquelin talked about active or passive strategies. However, the question we wanted to ask is there a place for bacteria as well? And we'd say, yes, there is, absolutely. It's very clear. Well, in that case, how would we sort of differentiate it? And as a general rule, we have this slide where we would see them being useful. Yeast is very effective in cold conditions, so it's perfect for things like cold maceration of red must. Particular strains like Pichia cluveri, and we have a strain called Fruitson of that species, is particularly good at scavenging oxygen. And so it's fantastic for protection against oxidative spoilage. In contrast to that, bacteria are great for preventing unwanted MLF, and very easy to use, very easy to dose, and potentially very good for protecting must when you need to transport grapes or even juice. A couple of other facets would be preventing high acetic acid, and we see a role potentially for both yeast and bacteria to achieve that end, of course, potentially through different mechanisms, and both of them have a strong place to play within low SO2 winemaking. So the next part of my talk is going to focus mainly on field trials, and I'm really glad that Professor Coquelin talked about validating what his group have seen in a scientific context in a more industrial situation, and that's exactly what I'm going to talk about. We have a couple of pictures here. This one on the left is in Australia last year, and it's where we actually added a product called NoVA Protect into receivable bins when machine harvesting Syrah grapes. And on the right-hand side, we have a picture where we've been inoculating some tanks in France with the same product during filling. So we've had this product NoVA in our portfolio for some time. Its actual name is, from using our culture collection nomenclature, is CHCC 12399. We've had the strain for 8 years. It's been commercially available, and we've always known there's been some quite good bioprotective effects. To help demonstrate that, we have the two pictures up on the screen. In both cases, we've plated either on the left-hand side, it's a wine sample, a juice sample. On the right-hand side, it's grapes that have been treated. But in both cases, on the left, we have the control, and as you can see, there's a lot of fungal growth on the agar plate. On the right-hand side is the one that's been treated with the NoVA strain. And although there is a little bit of fungal growth, it's significantly reduced. So we know that there's this fungostatic effect from the strain. But what about things like acetic acid bacteria or rogue Oenococcus that might cause an unwanted MLF during primary fermentation? Well, we've investigated that as well, and I'll just talk through this slide very quickly, but this is a challenge test done at laboratory scale. It was actually in a grape juice media with 1% ethanol added. And what we did is we took this juice and we actually purposely inoculated it with 1 million cells of Acetobacter oeni. We then had two further treatments where we inoculated Lactiplantibacillus plantarum at 1 × 10⁶, and then we incubated these samples for 3 days at 25 degrees C. Now the really important part is on this control one, we actually have outgrowth of 2.5 logs of Acetobacter. So the Acetobacter has absolutely flourished in the sample, which is unsurprising. What's really interesting is we have much lower levels in the 2 Lactiplantibacillus samples, and in the one using the NoVA strain, which is the one here with labelled CHC 12399, we actually see a net reduction of 1.5 log of acetic acid bacteria. So there's this very good bioprotective effect against a very, very common spoilage organism. But of course, that's at laboratory scale. Likewise, at laboratory scale, we've tried to do challenge tests to see if we can have a similar effect against rogue Oenococcus. And on this one, we've got it's a trial done with 6 different treatments. Again, it's a laboratory. We've taken some Aaron juice from La Mancha, and we've dosed with 2 different Oenococcus strains at 1 × 10⁵. So below what you would need for MLF to commence, but what would be representative of quite a high loading of adventitious Oenococcus. Now, we've measured here the malic acid at 3 days on the left-hand side and 14 days on the right-hand side. And the 2 bars on the far left here are the samples that have just been dosed with the Oenococcus. This graph is looking at the malic acid concentration, and what's clear is that the malic acid is consumed very rapidly with the Oenococcus present. In the samples with the same Oenococcus strains, at the same concentrations, where we've also dosed plantarum, what's lovely to see is we actually see negligible amounts of malic acid being consumed. So we've seen this really nice effect protecting the juice against this unwanted MLF. So taking things to the next level, this slide demonstrates a trial we ran in Australia last year. It was run in a commercial winery which is owned by a research institute, and we had 8 different tanks where we were able to try 4 different modalities in duplicate. The 4 modalities are where we've added either tartaric acid with the NoVA Protect strain or without the NoVA Protect. We have 2 more that have no acid addition, again with or without the NoVA strain. So it's 4 different modalities. We did add 20 parts per million SO2 at crush, so it's not SO2 free. And we did a fairly classical vinification. This is the fermentation graphs. Nothing unusual here, other than to say we gave a 1-day lag between dosing either acid and or Lactipantibacillus plantarum, and then adding yeast. One of the great things in Chr Hansen is we as a biotechnology company, we have some really phenomenal resources in terms of biochemical assays. And one area we're able to utilise in our wine team is qPCR. PCR is quite interesting because before COVID came along, the average man in the street probably had no idea what PCR stood for, but now everyone does. Nevertheless, PCR is a tool that we're able to access, and we believe it's a tool that's it's got a lot of potential within wine microbiology. So to talk about this slide here, there's a lot of data here. On the left-hand side here, we actually used PCR with a very specific marker, which was strain-specific for the CHCC 12399 strain. And what we used that for is to demonstrate implantation. So we wanted to prove that the tanks of wine that had been inoculated with the Lactibacillus plantarum actually had it there. And the great thing to see is, yes, we were able to demonstrate that really effectively. There's actually 4 points that were measured. If it's not clear, the first one, which you can't really see on this first graph because it didn't pick any up, is actually at day 0 before inoculation. Then we have day 0 after inoculation, day 2, and then at the end of alcoholic fermentation. And the really good thing is we're able to see plenty of Lactiplantibacillus plantarum in the the 4 tanks, because it was in duplicate, that were inoculated, and none in the others. So that's good. We've demonstrated implantation. But on the right-hand side, we've got another picture, which is actually very, very interesting. And that is where we've used qPCR with a generic marker. So it's species-specific, not strain-specific, for Oenococcus oeni. And along the bottom here, we can see if we have the first sample, that's with both the tartaric acid added and with the Lactiplantibacillus, and we also have just with the tartaric acid, we see a very similar picture. And actually, tartaric acid's quite a good biocontrol agent, and we see exactly that. The amount of outgrowth of Oenococcus is restricted. If we look at the far right, the third one along, we have the sample which was dosed with the NoVA strain, but without any acid or any NoVA. And what's really interesting is we actually see a lot of outgrowth of Oenococcus happen. Conversely, in the last combination we have here, we have both. We have the NoVA without any acid. We see as good as, if not slightly better, bacteriostatic effect from the NoVA strain in relation to tartaric acid. So we think that's actually quite significant. We've shown something that works better than tartaric acid addition. Of course, with any winemaking, the end game isn't to demonstrate prevention of spoilage. The end game is actually to make a good wine. So with these wines, we put them through a sensory panel in France towards the end of last year. In fact, we didn't put all 4 through, we chose 3. We put through the control, which had no acid and no NoVA. We put through the treatment, which just had tartaric acid. And then we also put through the treatment which just had the Bioprotection. And what's really interesting is there is significant differences in the wine. They all taste like Cabernet Sauvignon from the Grampians region, but there's greater fruit weight. There's more red fruit emphasis on the wine that has the NoVA. On the graph there, the grey line is the NoVA, the blue one is the tartaric acid treatment, and the orange is the control. So that was really interesting. The other really cool thing, this is Cabernet from a reasonably cool region in a cool growing season, so there can be a propensity towards green flavours. We actually have a very non-green wine, so we're able to mask that greenness. In the mouth, we also see some really good effects. We have less bitterness and better overall balance and persistence in the wine with the Bioprotection. So that last trial was carried out at fairly small commercial scale. What happens if you take it up another level again? And this is an interesting example from Tuscany from the 2021 vintage. And it's from a cellar which has a particular problem with unwanted MLF during primary fermentation. And the picture they typically see in their winery is this. On this graph, we have cell count along the y-axis, and along the x-axis we have the time. And this green line that suddenly goes from low to high is the concentration of Oenococcus. And as you can see at day 4, it suddenly ramps up. The result of that is this picture here. And the green line in the second graph is the malic acid concentration. And as you can see, MLF starts rapidly, and we're in the middle of alcoholic fermentation, so it's not a very ideal picture. Taking an equivalent tank of must, we see this picture. So we've represented the Lactiplantibacillus inoculation here on the graph. But the great thing is we don't see the outgrowth of the Oenococcus, which is represented by the green line. There's a little bit of growth, but doesn't go anywhere near high enough to cause an unwanted MLF, which is why we see the picture on the right-hand side where we see a little bit of malic acid degradation. I would say that's going to be down to the yeast, but we don't see an MLF. The other nice thing is we have a lower VA in the finished wine, 0.55 versus 0.62, which also ties up nicely with the inhibition of acetic acid bacteria, which both Professor Coughlin and we have seen it at a laboratory scale. But that's not the only advantage. If we actually look at the fermentation kinetics of the two different tanks, on the left-hand side we have the control, on the right-hand side we have the one treated with the NoVA. As you can see, it's a much more even, steady fermentation. It finishes 1 day sooner, and there's the lower VA we talked about. The winemaker himself was very happy with this, and as he said to us, this wine is clearly cleaner than the rest. When tasted, it is less oxidative, cleaner than the other tanks, the fruity note stands out, and the tank is amongst the best in the cellar. Great job, and I would say useful for everyone. So that's great when we launch a product to have feedback like that and to see it work in a real life environment. But taking things one step further, and this is my last set of slides, this is actually from an extended application of our NoVA Protect product last year in France at a cave cooperative called Cellier Charles Cros, which is in the Corbière region. It's a very interesting winery because they have both their estate labels, but they also trade a lot of wine to negociants. And for them, they get a much, much higher return by selling a preservative-free wine compared to a conventional one. So there's a strong emphasis on being able to make a good, clean, preservative-free wine. So they had a program of making 8 tanks preservative-free last year, 7 of which we inoculated with the NoVA Protect. We have a lot of data from this trial, but what's really nice to see is the one tank which has by far the highest volatile acidity is the one tank that we did not inoculate with the NoVA Protect. When we look at the wines, and we did qPCR on this one as well, we saw very, very good implantation on all of the wines, except of course this one here in the middle, tank 707, which was not inoculated, and there we have negligible amounts of Lactiplantibacillus plantarum CHCC 12399. We also applied the same analysis we did on the previous trial as to looking at the Oenococcus, and while none of the tanks did have a high level of Oenococcus during primary fermentation, which is great, we did tend to see the highest increase in the tank that wasn't treated with the NoVA product. But the most interesting thing, I think, is the acetic acid bacteria counts. Again, we did qPCR. Of the 8 tanks, the only tank where we saw an increase in acetic acid bacteria from day 0 to day 2 was in the one tank that we did not inoculate. In every other tank, we can see a very nice drop in the acetic acid bacteria levels from day 0 to day 2, which is exactly what we wanted to achieve. So that brings me to my conclusion from this trial. We saw clear implantation. We saw the acetic acid development happen the most in the tank that wasn't treated. We linked that back clearly to the fact that there's more acetic acid bacteria in the untreated tank. And the other thing, it's pretty hard to represent, but the feedback from both the winery and the enological consultant responsible for the wines were that these wines graded extremely well. So the quality was there as well, which at the end of the day is what we're trying to achieve. So this is my last slide. It's just to conclude, and it's to really introduce our new product, Viniοποura NoVA Protect, the first bacteria exclusively for biological protection in wine, or musts. It's a very easy to use format. It's for direct inoculation, so it's very, very flexible. It can be added any time from once you pick a grape to when you crush the reds or fill a tank. It's a really convenient usage rate. It's 1 bag per 25 hectolitres, or if you're adding to berries, per 3.5 tonnes. It gives an inhibition against adventitious Oenococcus that could cause an unwanted MLF. Gives inhibition against acetic acid bacteria. It's a strong alternative to using either high levels of sulfites or to acidifying musts with tartaric acid. And it can be used very nicely in low SO2 winemaking, but not exclusively so. It also has a place in conventional winemaking. So thank you very much for listening, and we're going to open it up to questions now.